the collection of Humanini at the Natural History Museum of Venice.
Hello, everyone. I'm Davide Del Posa. I'm a PhD candidate at the University of Central Florida, where I'm studying different aspects of ichneumonic morphology. I was supposed to be at the ECN meeting this morning, but unfortunately, I'm in Florida right now, and due to this big storm that across Florida and especially Central Florida, they canceled my flight. They rescheduled it for Sunday, and I couldn't be there. So I'm very sorry. I was hoping to meet you all there, but this is the way. It is what it is, and I still wanted to give this talk, and I'm recording it, so sorry for any mistake. But essentially, I want to give you an account of a small collection, not big as the Smithsonian or the American Museum, of a small collection at the Natural History Museum of Venice, which is, because of the number of specimens and the number of type, it is probably one of the largest for human eye, all right? It is the Giordani-Soica collection. So I will start immediately giving you where is located that collection. So right now you are in Vancouver. Of course, I'm in Florida. Okay, I was supposed to be there. But the collection is located in Italy, which is across the Atlantic, and specifically in Venice, that many of you will know for touristic purposes, but probably only a few of you know that there is one of the biggest natural history collection in Italy. Historically, is beautiful. It has fantastic entomological collection, but also from an historical, architectural perspective is absolutely amazing. And Marco Liana is the current curator, is working with Beetle, right? Within this collection, within the Natural History Museum, you have one of the biggest collection, as I said, of vespids. It is called and still remains with this label, the Giordani-Soica collection of vespids. Who is Giordani-Soica? Giordani-Soica is this distinct gentleman, all right? Now, it's the only picture that we have. Back in the day, it was in London, right? That worked in the museum as a director between the 1946 till the 1978, which is the year that he retired completely from the museum. His activity, though, spans well before his activity as the director and well after his activity as a director. And this is one of his first publication on the vespid. Despite him doing a little bit of work on other groups, some Coleoptera, some Diptera, and also some amphipods, his core of his activity, it was a taxonomist, of course, was on vespids. And specifically, he described a lot of humani and introduced more than 102 genus level names and more than 1,346 species level names. This is an image of his, one of his publications was done by his wife, Gioiella d'Este, right? When he retired, he decided to start selling his collection to the museum. So there was a little bit of internal conflict because he was director, he was having his own private collection, and now after retirement, he was selling it to the municipality, which was in charge of the museum back in the days. The acquisition of this collection from the museum was done into three major steps, which are important to understand how the collection was handed over and inherited then by Marco Liana. The first one and the largest portion was in 1978, with more than 387 drawers and more than 28,000 specimens. The drawers, though, were the small drawer, the Italian way of doing, sometimes the former way of doing stuff, and were these small black boxes, sometimes without any glass on top. So you needed to open to see the specimen within. And to give you a perspective of the dimension, they're extremely small, not as big as the American. And you can see they're small because I'm a big guy and beautiful, but very big guy. And you can see how small they are and how internally they are organized. The second part was acquired in 1995, okay, 2 years before his death, and only comprises 50 drawers and 1,400 specimens. And the last part of the collection was donated after his death by his wife, Giuliana d'Este, with more than 148 drawers, more than 10,000 specimens, 200 holotypes, 4 lactotypes, 75 allotypes labeled as allotypes, and more than 1,240 paratypes, right? The organization internally was this. You usually had specimen with, eventually, if you got the type, you got the red label underneath, and then you have the header label to identify the species or the genus that was coming after it. There is one point to make. We're not sure of how, across these 3 steps, of how many specimens were resold to the collection, because during his retirement, he kept an office at the museum, and we think that probably he brought some specimens from his first acquisition back to his own private collection and resell it again in the second and possibly the third acquisition. So the condition upon acquisition was that we had 480 drawers and 12 cabinets. This is what Marco and then later on myself saw when we got to the collection. And you could see that could be sometimes the cabinets were almost empty or half and half, or fully, like in this case, fully full of cabinet of drawers, or sometimes we have also different boxes, sometimes not labeled or not labeled correctly. You can imagine how difficult it is to buy such a large collection in such a bad condition, let's put it this way. Of course, the specimens, though, were enormous, 20,000, more than 20,000 species, more than 2,000 species, the types 3,000, of which 450 holotypes at the end of the day. This was the first assessment that Marco did when he came into work at the museum. And also the internal organization was fully organized, like in this case. Sometimes you got the drawers that were almost empty. Sometimes the condition of the specimen were not so great. You didn't have any header labels. Sometimes you see those oranges patches. Those were for liquid, where the genitalia were embedded, and you can see that they crack off and they were accumulated on the side of the drawer. And you can see that also could be extremely messy, because what Giordani Soica was doing is sometimes storage genitalia and specimen in 2 different boxes, giving them the same number or same symbol, and that you need to match it. But you can see that when the genitalia crack off and accumulate, that becomes a huge problem. So when I entered as a volunteer into this collection, there was a need for a sort of reorganization, restoration of the collection, and improvement overall. So we decided to start. Little we know that it was a huge amount of work. And we divided this work into 4 phases. Some of these phases were not clear-cut, but it helped that given this sort of talk. So the first phase was simply a database, and we use an internal database developed by Marco, Liana, in which we keep track of the species, the number of specimens, the number of male and females, and their placement. And we put them back into the same placement again. There was just a count of the specimen, right? And this took about 3 months of full time. If I need to split up because back in the days I was simply volunteering, so I was studying and finishing up my master. So we did this probably in 1 year of part-time job, but 3 months of full work, right? The second phase was probably the funny part. At this point, I got a job within the Natural History Museum for about 1 year. And it involved the actual physical reorganization of the collection, which means that we went back to the database, checked the species, found the specimen, understand if the species were located into different boxes, and we moved the specimen into one single box, or the same the specimen belonging to the same species. We put it all together in order to reorganize the collection and saving some space. We also, in this phase, restored the genitalia. We'll talk a little bit about the genitalia in a moment. And this took the longest period. This one was probably more than 6 months of actual work, full-time work. And of course, you can see my frustration after a while understanding what Soika did, especially in some of the boxes. Like this box here, where you can see the genitalia were completely cracked off. So what did we do with this one? Very easily, we took all the pieces on one box, and we tried to match, all right, the borders of them, the corners, to see if those are belonging to the same type of group of cracked liquid. After that, we put it into water because this is for liquid and it's water soluble. After that, we took only the specimen, we took only the genitalia and put it into another liquid, DHNS, which is a transparent liquid that after a while becomes hardened, okay? And we place it onto a label, and the label we place it underneath the specimen if we were able to match the specimen. Of course, you can see how this could have been extremely tough, because in this case, it took me several days to understand what pieces were all together, all right? After this huge phase, we started the third phase that was sort of concomitant, but we also did it separately even later. We re-database everything. So we went back, we double-checked our collection, we re-update our database with the latest collection number. And also what we did is we take the primary types and we check them against the original publication. We wanted to see which one were correctly stored into our collection. And if in the collection were, say, tipo nella mia collezione, which means types in my collection, at that point we know that we were legally the correct repository for the types, all right? And this took about 1 month. It was the final part of the restoration. The last one, the fully last one, was the fourth phase, which comprises the labeling of the specimen, the drawers and the cabinets, inputting the header labels, like you can see here. This is the final part of the work. You can see that we organize it by genera, and within the genera, we went across alphabetically with the species. So we have a header label for the genus and a sort of initial label for the specimen to go across, all right? You see, beautifully organized, easily to access. We also, this took about 1 month. As I said, we also label the cabinets and the drawers. This is easier, and we divide the drawers by geographic area. It was easy for Marco access this way because a lot of people ask for specific areas, at least in Humana. So the problem that we encountered, some of them you already saw them. Well, we have the problem with the genitalia reconstruction, and not just matching the different pieces together, but also matching the specimen to the genitalia. And for some of these genitalia, we weren't able to actually match the specimen. Sometimes there are at least a couple of holotypes that we're not sure where the genitalia went. We have same species in multiple drawers, so we have multiple specimen of the same species in multiple drawers. We have Thai specimen missing, and we have Thai specimen from other museums that are in our collection, but they were not supposed to be in our collection, even historical stuff. And we've found also specimens that were labeled as types that are not actually types because the specimens were collected after the original publication. And we have also variation among this. What is an allotype? Sometimes he label allotypes in a weird way. And so we maintain that, but there are problems that possibly future taxonomists can resolve now that the collection is fully indexed. The results, however, is that we were able to save some space. Now we have only 246 drawers. Before we have more than 430. Now we have only 7 cabinets instead of 12 as before. Drawers, we found drawers from other museums that possibly were loans given to Giordani Soica, and we found about 45 boxes of these, which is possibly 1 cabinet of those that you saw before. And we found 50 holotypes belonging to other museums. And we restored about more than 200 of those in Italia. And this reorganization helped other taxonomists, not ourselves, to describe more than 34 species. And to what I understood, there are many more to come, thanks to this reorganization. You can see the final organization, and this is the division that you see in biogeographic area. So we have 75 boxes for the Palearctic, 63 for the Apotropical, 41 for the Oriental region, and so on and so forth. So we have specimen and we have a collection that is worldwide and extremely of interest, especially the Australasian. We have a specimen from Australia. They are known only for Thai specimen, and we have that luckily. This one led us also to be able to complete after 6 years from the starting of the project. As I told you already, little we know how long it take it. We were able to publish it in Zootaxa. We published the appendix, 2 appendices, one for the checklist of the species, one for the actual Thai specimen. Please, if you're studying this, give it a read, send me an email, and I will be more than happy to send you the link. And I think this actually is the best end that we could accomplish with this collection. There's a lot of stuff to do, and we hope that this publication will help foster new studies of this collection also for the future. With that, I'm finishing up my talk, acknowledging the people that across these 6 years helped us, or helped me specifically. Alberto Ballerio for the some nomenclatural issues, assistance. Marco, who is an Italian taxonomy working on Whisenant specifically, who help us identifying 2 specimen or giving a better account of the collection. Gavin Broad, that helped us understand what type specimen belonged to him and what belonged to us. Giacomo Masato was the staff member that helped us with the bibliography. I want to thank also Mark Uliana and James Carpenter. They are my co-authors, and without James Carpenter, we couldn't have been done this work because of the amazing papers that it did over the years, and he gave us also a lot of unpublished material that we could actually work on the collection. And of course, my big thank to the entire staff of the museum that welcomed me at the museum for those 3 years that I worked there as a volunteer first, and then as an actual part of the staff. And with this one, I thank you all for listening. I'm sorry for all my mistakes, and I'm sure I did a lot. And I wish you the best for the future of this meeting again. And I really hope to meet you all on the next meeting in Maryland. And I apologize again for not being there. Thank you, and I'll see you later.
I got
That's it.
screen over there. No, unfortunately.
All right, thanks so much to Davide for that talk. I apologize for going over time, but I'm also really impressed with him being able to pull that off upon hearing his flight was canceled. Our next presentation will be from Andy Austin, who's come all the way from down under to tell us about Taxonomy Australia and its mission to document the continent's biota.
Good morning, everyone. This presentation doesn't contain a huge amount of sort of insect systematics, but I hope I'm going to be able to tell you a few things that are going on in Australia over the last few years that have quite significant and hopefully successful ramifications for both funding sort of insect and systematics on other groups and also for collection infrastructure and other things. So I hope there's something of use for at least some of you. It's fair to say that historically the Australian systematics community has been really siloed. Traditionally there's been a separate systematic botany society. There's a general systematic society, which by default has become a sort of faunal systematics society because the botanists sort of have their own group, and also the mycologists, systematic mycologists also have their own group. And it's been that those groups in themselves are successful, but it's limited the ability of the taxonomic community to advocate for funding to both federal and state governments. And that's sort of held back the sector quite significantly. So what's happened is in about 2016, following discussions between the three systematic societies, a plan was formulated to develop an overarching organisation which was called Taxonomy Australia, and it was to represent the taxonomic community for the country with a view of supporting and expanding sort of taxonomic science. We were able to garner some funding from the Potter Foundation, and most significantly, the Australian Academy of Science got behind this initiative and provided both initial and ongoing support in various ways. And one of the things that funding was used for, it appointed a projects officer for 2 years, Kevin Teely, who's a botanist who worked at the herbarium in Perth, and he has an interesting skill set that made that appointment really ideal. And the other thing that happened was the Australian Academy of Science was keen to work with Taxonomy Australia to develop a decadal plan, which is essentially a strategic plan for the sector. It was called the Decadal Plan for Taxonomy and Biosystematics in Australia. So let me tell you a little bit about decadal plans. So the Academy of Science has a program of developing decadal plans for major sectors of the science community in Australia. And so they're really strategic plans for the operation and development of science sort of over, a 10-year period. And they've been used by some of the groups, in particular the astronomy community and the geoscience community, really effectively to obtain significant increases to the funding base for both competitive and non-competitive research funding. And so with the collaboration between the Academy and Taxonomy Australia, we developed a decadal plan entitled Discovering Biodiversity, a Decadal Plan for Taxonomy and Biosystematics in Australia and New Zealand for the 10 years from 2018. Fortunately, the New Zealanders haven't really come to the party as effectively as they should have, and so it's kind of fallen back to be a national decadal plan. But within that, there are sort of 5 key elements which really become the major aims for Taxonomy Australia over sort of the next 10 years. So the first one is to significantly accelerate species discovery and documentation using a range of new and emerging technologies. to integrate and enhance sort of taxonomic infrastructure, and this particularly relates to unifying what is sort of a dispersed sort of array of biodiversity collections, and to develop them into sort of a distributed science infrastructure. Support strategic growth of taxonomic capacity and capabilities, and primarily that's an expanded training program at both doctoral and postdoctoral level and beyond, to enhance the services for end users, utilizing new technologies for both data management and also enhanced diagnostic tools, and importantly, to engage the community, including Indigenous Australia, into helping to document the biota and recognise its uniqueness and its value. In association with this, and the Australian Academy of Science also were kind of insistent and talked us into working with them to develop an economic statement about the value of taxonomy to the national economy. And so the Academy appointed Deloitte's Access Economics, which is an independent economic consulting company, and they provide sort of economic services that include sort of economic forecasting, modelling, analysis, and advisory services across the spectrum, in particular to help sectors of society, including non-government organisations and even private industry, to really navigate successfully the complexities of government economic policy. I'm sure your economic policies in this country are just as bad as ours, and it's a minefield. And so having a group like this to be able to sort of advise was really important. So this was commissioned by the Australian Academy of Science. It's a very dense 100-page document, of which I have to admit to understanding not much more than the executive summary in the first 3 or 4 pages, and then it gets into the detail of the economic analysis. But that's what they do, and they do it really well. Take-home message from this document is that for every $1 invested in taxonomy, it'll bring between $4 and $35 in benefits to the economy, including benefits to biosecurity, agriculture, fisheries, environmental assessment, and so on. And this is particularly important if we're going to go and talk to politicians about, in an advocacy process to increase the funding base for taxonomy in the country. So the governance of Taxonomy Australia, there's an executive, Jane Melville, who's a herpetologist at the Victoria Museum in Melbourne, that chairs the steering committee, and with Tom May and myself, we form an executive to represent Taxonomy Australia in whatever advocacy processes we engage in. And then there's another 12 people on the steering committee, which pretty much covers the whole of the taxonomic community in Australia. The 3 systematic societies, universities, the heads of the Council of Australian Faunal Collections and Herbaria, and a number of other key groups, including Atlas Australia, the relative government departments, and so on. And that group meets online about once every 6 to 8 weeks. So just to sort of step back and give you a picture about the progress that's being made sort of in describing the Australian biota, on average, only less than 1,000 species are described for Australia every year. And the breakdown of the major groups looks like this. About 44% are insects, 62-63% in total are arthropods, 15% are vascular plants, 10% are fungi, and so on. And then the minor groups sort of make up about 22%. But the total biota, there's about 160,000 described species for the continent. We're talking about the groups that we've focused on with are essentially eukaryotes. We haven't included microorganisms and viruses in this analysis. So we think about 30% of the Australian biota is described, and the estimate, which is a hand waving exercise, really is just over, we think, 500,000. But look, the elephants in the room are the fungi, nematodes, and the mites. And the number of systematists in Australia that work on vascular plants, there's about 5 times more vascular plant taxonomists in Australia than there are who work on those 3 groups. In fact, there's virtually no taxonomic effort on nematodes in the country at the moment. There's very little on mites. And so you take those groups and the hyperdiverse insect groups and several of the marine invertebrate groups, in particular polychaetes, and there's very much a mismatch between resources and what is going to be required to tackle, the really big hyperdiverse groups. So the key issue in all of this is to substantially increase the funding base for taxonomy. And if you asked me 12 months ago, I would have thought that the chances of pulling that off were probably, pretty slim. But there's several things that have happened just in the last 12 months. Okay, so there are 3 things. And so the first one is we've had a change in government. The Liberal coalition government, which is a right-wing government, were in power for 9 years. They disbanded the Climate Change Adaptation Research Facility. They reduced funding to conservation by 40%. There was static funding into this sort of the whole research sector in Australia, with the exception of medical research. They got rolled big time in the election this year, and the Labor government is kind of going out of their way to dismantle some of the things that the Liberal government did, particularly in sort of climate change adaptation and sort of a whole range of related areas. The other 2 things was that this year the State of the Environment report was published, which I'll say a bit about in a moment, and also the government department where the Australian Biological Resources Study sits have initiated a major external review of that operation. So for those of you who are not aware of it, ABRS is a competitive grant scheme that exclusively funds taxonomic research on all groups of organisms. And it's probably one of the few granting agencies that solely focuses on taxonomy anywhere in the world. And it's been instrumental in funding and supporting sort of postdoctoral and doctoral research, as well as major taxonomic projects for a very long time. In fact, it has its 50th anniversary next year. So a little bit about the State of the Environment reports. So these are government funded, but they're developed completely independently by a group of specialists. There's a State of the Environment report every 5 years. So the one for the 5 years up to 2021 was released in August this year. The take-home message from that is that the health of the Australian environment is poor, and that everything points to further deterioration in sort of coming years. And that would be the same for most places, I guess, around the world, wouldn't it? The interesting thing this time is that for the first time, there are several statements in the State of the Environment that directly relate to taxonomy, including the inadequate level of funding provided for taxonomic research, reduction in the number of taxonomists employed in museums and herbaria is a key factor about a reduction in the number of species that are described and the tools that are developed, and an over-reliance on retired and honorary researchers, which, having just retired last year, I kind of take that as that's a positive as well as a negative statement. But also it makes a specific statement about the Australian Biological Resources Study, which runs this National Taxonomic Research Grants scheme, that its budget's been fixed for the last 10 years, and with no indexation at all, its ability to support the sector has declined. And so the situation now is that we've probably, with some of those ducks lining up, we've got the very best chance in the next 12 months to advocate for a significant funding increase through the ABRS for taxonomic research. And Taxonomy Australia is going to bid for a fourfold increase in the budget of the ABRS, as well as a number of other initiatives. And so through the Australian Academy of Science, we've been able, or in the process, of organising a meeting with the Minister for the Environment. And that's probably the first time that the taxonomic community has been able to even meet with someone that's that highly placed in government. The fact that they're prepared to meet us is overall a good sign. If they weren't sympathetic to this, they would not meet with us at all. Also, Tanya Plibersek, the Minister for the Environment, happens to have a major interest in native plants. And so she has some understanding about what it is that we're trying to do. So look, that's, I think all I wanted to say. Two things: if you're interested in having a look at some of this, the website for Taxonomy Australia is just www.taxonomyaustralia.org.au, and it's a very, very slick website we had access to the media and graphics department of the Australian Academy of Science in putting that together. And if you're interested in the Deloitte's, following up on the Deloitte's report, if you just Google Taxonomy Australia Deloitte report, you'll find it straight away. Thanks very much.
I want to ask you questions about that later, but I'll track you down after we're done. Thanks so much, Andy. We started about 10 minutes late. I'm going to try to keep it to exactly 10 minutes over time until we can recoup with our break. But I'm excited to hear about our next talk with Ernesto Recuero. How well can we identify soil and leaf litter arthropods using DNA barcoding?
Thank you.
Hi, everyone. I'm Ernesto Recuero, currently a postdoc at Mike Caterino's lab at Clemson University, and that's what I'm going to talk about. As we all know, we are far from knowing the real diversity of life on Earth. There are millions of species yet to be described, and even the ones we have already described, for most of them, we barely know nothing. And this lack of taxonomic knowledge is being called a taxonomic impediment that is largely caused by the shortage of expert taxonomies in so many groups. This makes identifying a species a difficult task in many cases. And this is not only a taxonomic problem, it's a problem for other research disciplines, since if you cannot identify an organism, it's very unlikely that you will want to study anything about it. So DNA barcoding was intended to help people identify species without any taxonomic training. It's based on the association of DNA sequences with particular species and the use of reference databases to compare your data. And it assumes, it's based on the assumption that the intraspecific differentiation is going to be much lower than interspecific differentiation, forming what is called the barcoding gap. Although this is not always so clear. For instance, these are some data from Symphypleon springtails from the Southern Appalachia. As you can see, the barcoding gap is barely formed, and there are a lot of very highly divergent pairwise distances. What's interesting about soil and leaf litter arthropods, they form very diverse communities. You can find tens or even hundreds of species in a single sample. You can find representatives of almost every major terrestrial arthropod groups. You can find different ontogenic stages in your samples. So at the end, it's hard to put a name on every specimen you find for a single person. This particular work is based on a small subset of data from a much larger project trying to characterize the diversity of litter arthropods from high elevation forests in the southern Appalachian Mountains. If you're around on Wednesday for the ESA meeting, you can hear from Mike Caterino talking a little bit more about this project. Sampling followed standard procedures. You just sifted litter, extracted the specimens using burlesque funnels, and did some time-consuming but very fun specimen sorting. We tried to isolate morphospecies from each location and selected one specimen from each morphospecies to be processed. That specimen was photographed to generate a digital voucher, processed in the lab to generate the barcodes, and finally they were prepared to be incorporated in the collection. So at the end, we wanted to have one physical voucher, one digital voucher, and an associated barcode. If you are working with just a few samples, a few barcodes, it's quite easy to analyze the data directly. But when you are working with hundreds or thousands of barcodes, you want to use some automated method that in a few minutes can give you some ideally correct identifications to most of your samples. So we have tried different databases. Midori eukaryote-1 reference are fed with GenBank data, while Bolt has its own data source. Although both of them, both GenBank and Bolt, are continuously exchanging that data. We try different informatic methods. And at the end, we wanted to check whether the obtained identifications were correct or not. So we try to identify morphologically our samples to the lowest taxonomic possible. And whenever we can get to genus or species, we just blasted them in GenBank and against the proposed taxa. And if the identity percentage was above 95%, and we consider it a correct identification. If it was below that, we consider it an incorrect identification for a species with an 80% threshold for genera. So we started with around 750 samples, for which we obtained almost 500 barcodes. Some groups did very well in the lab, like Collembola and Coleopterans, while others perform quite poorly. Hymenopterans, we get almost just 1/4 of the samples amplified and sequenced. This is probably because the primers we have been using are not working well with this group, but also maybe there are some inhibitors in the DNA extraction from Hymenopterans. So we have to improve this a little bit more. And the identification results were extremely disappointing. Most of our samples could not be identified, as the red part of the columns are unidentified samples. Just a few samples could be identified to a species or genus. The best performing method was Bolt. Around 14% of samples could be identified to a species, and 15% of samples could be identified to genus. Also, at the species level, we got some incorrectly identified samples with all methods, which is probably the worst thing that could happen. We probably would prefer to have unidentified samples than incorrectly identified samples. We observed some differences among groups. The spiders were by far the best performing organisms in our sample. We could identify more than 50% of our samples to a species and around 60% of them to genus level, with very few incorrectly identified samples. So not so bad for spiders, but springtails were a total failure. No sample could be identified at the species level, and we just identified one single sample using one of the methods to genus. So no good at all. Other groups, as beetles, had intermediate results, a few identified species, almost 19% of the samples identified to species with BOL, more than 20% of samples identified to genus, but again, with quite a few incorrectly identified species. So the conclusion is that these methods are totally useless for identifying our species, at least in our case, in the study case. The main cause for this is that the species we are finding in the Southern Appalachians are not represented in the databases. The differences among groups, for instance, spiders that did relatively well, probably they are better studied group. And also, it seems that they have some widespread species. In fact, most of the species we identify among our sample are also present in Eastern Canada. That is a very well characterized region for DNA barcode studies. On the contrary, Collembola are probably much worse studied. They probably include many narrowly distributed species. And also they have this extremely high genetic differentiation even within species. So that can affect the performance of DNA barcoding. In these cases, maybe using a different gene with a lower mutation rate maybe could improve these results for these particular groups. But there are other problems that are making us getting incorrect identifications. For instance, incorrectly classified barcodes in the databases. We found some larvae of these fly species in our samples. If we try to identify the barcodes using BOL, we get the name Nearticopus canadense. While if we use gene bank based databases, they are identified as Nearticopus pecki. This is curious because in both databases, they are using exactly the same sequences. These are shared sequences among databases. So the answer to this question is that BOL is much more actively curating their database. They are revising identification, actualizing taxonomic information, while taxonomic actualization in GenBank takes forever. It's sequences can remain there for years and years, incorrectly identified, and it's very hard to correct that. Another issue we found is when we find very low interspecific distances. For instance, trying to identify with barcodes sequences of Geostiva nibuligena, the species on the left. We always obtain with all methods and all databases identifications as Geostiva bicarinata. Well, of course, there are no representatives of Geostiva nibuligena in the databases. And if you see here, the genetic distances with Geostiva bicarinata are very, very low. So this is within the range that is considered intraspecific differentiation. So every method we've tried will identify the barcodes of nibuligena as bicarinata, getting an incorrect classification. And well, the barcoding can be useful, but there is still a lot of work, a lot of taxonomic work to do if we want to make it really useful for everyone, for every group, for every region. And collection can play a major part in improving these databases, since the material, the most important part for this is the material, the specimens, correctly identified species. Collections have that. So either by producing barcodes and uploading them to databases, or collaborating with some initiatives as BOL to generate those barcodes, can make this tool really useful for people that need to put a name on samples. Thanks for the institutions that made this project possible, and thank you very much.
Get that.
All right, I apologize for not leaving much time for questions. I think we're going to go ahead and try to get on to the next talk to get back on schedule, but I highly encourage you to track down Ernesto and Mike and Frank and everyone else who's been working on that project. I know we all probably have a lot of questions about DNA barcoding, leaf litter arthropods, the best arthropods. Our next talk is entitled Wall of Insects, Lessons Learned in Muraling from Jacki Whisenant.
Hey there, everyone. I am the reason that we are 10 minutes late, and I'm so sorry about that. But thank you all for being here, and I'm really excited to talk to you about this in-progress mural for the Wisconsin Insect Research Collection. I'm so excited.
It should be good.
Okay, great. So the whole motivation, I'm going to kind of talk through this pretty quickly so we can get back on track. So the motivation for doing this mural was to highlight our insect research collection, which is a wonderful hidden gem on campus. And twofold, we want to create an educational poster that can be for sale through the UW Zoological Museum store, which is set up to create posters for highlighting different Wisconsin fauna, and has been a decent sort of fundraiser for the museums on, or this museum on campus. And the profits for that, from that poster, will of course go back to the WRC. And it's also just a great excuse to draw a bunch of insects and give back to a collection that has literally changed my life. One of my main inspiration was my teacher and friend, Jane Kim, who is responsible for the Wall of Birds at Cornell. She, as part of her internship for her science illustration program, that became a 2-year extended project through the Bartell Science Illustration internship in Cornell, where she made a massive wall mural and kickstarted her career of doing extensive mural projects, founding Inkdwell, which is a studio that has recently celebrated its 10th anniversary and continues to make massive murals across the United States. So drawing upon that to let's talk about Wisconsin, Wall of Insects. We will actually be co-presenting at Spinach next year. So if you're in the Cal Academy in May, please come and see us. We love to talk to you about muraling, and she'll mostly talk about muraling because she's got the experience, but I'll be there and be like, me too. So this was a conclusion of the California State University Monterey Bay Science Illustration Program. And this is, that program ends with a 10-week internship for every student at an institution of their choosing. And this was my choice. And we're ready, fall of 2017, let's go, let's put some bugs on the wall. And there was construction. So the HVAC system was being upgraded in the building, which is very good to do. We all want to breathe, it's fantastic. But that necessarily delayed the start of this mural for a few weeks, and then a few more weeks, and then a few months. And then the hallway was finally free in spring of 2018, when I had already started my master's with Dan Young. So during that time, I was like, okay, I can plan, we can do this. So put together a list of Wisconsin families, choosing ones that were commonly encountered, using occurrence records on Bug Guide, iNaturalist, and in the WRC collections themselves to say, what are people encountering? What are they finding? What would be good to be sort of a representative of this family that would be familiar, and then also draw people in. It's like, I've seen that before, that's great. All insects are scaled up to about a 10 times adult life size. And total, there are 330-ish insects on the wall. Some of them are duplicated, so like showing a male and female example. Some are paired together as gynangiomorphs, just for space efficiency, especially with those darn butterflies. And so we have that kind of going. I will kind of motor through these first introductory slides. We're just going to get straight to the pictures again, so we can get back on track. But I'll talk. I'm showing them so that you can go back and take a look if you want on the recording. So for the design work, the normal process of working with an artist and making a design for anything is you sketch, and then you do kind of a color draft, and everything's okay at that point. Then you proceed with the final artwork, and ta-da, it's great. The mural kind of it proceeded a little more organically instead of designing the whole thing, which in hindsight would have been a great thing to do. I went sort of insect by insect, sketching them first to decide their pose and figuring out where they would sort of be on the wall before physically putting them on the wall, paint them in primer, black and white primer, and then color glaze over. So looking at this initial layout, we have a phylogenetic tree being all fancy and spiral, whatever the spiral geometry fancy thing is, laying out tape around the tape so that you can have really clean lines. I used to be a motorcycle painter. That's kind of like our clean line thing. So we're just bringing all sorts of background experiences into this wonderful wall. And then it was like the first set of insects that I did was draw them on paper, scale them up on the computer, print them out, tape them on the wall where I wanted them to be, transfer them, and then finalize the sketch because transferring looks like crap. And then I was like, wait a minute, let's just draw them on the wall. Let's just measure them out and draw them on the wall. Skip 3 of these steps. That sounds fantastic. Which also allowed me to adjust for optical distortion. So when you're standing 3 feet away from a wall or 2 feet or 4 inches, when you're looking down at the far bottom and then the far top of the wall, you're having, you're experiencing this sort of optical stretching of what is painted on the wall. And that's, that was a little bit unnerving at first. And I had to be like, oh no, I swear I know how to draw insects. They don't look weird like that, I promise. And so that was kind of an adjustment to make in that sketching on the wall stage that I wasn't really anticipating. Style choices. There are certain, I would highly recommend using a simpler style because that would be great. So I went with like the top 2 kind of options because I was fresh out of my science illustration program and I'm like, I want these to be so detailed and they're gonna be so great and I'm gonna feel so fancy. But honestly, please, when you're considering murals, go with something a little bit simpler, a little more manageable, a little bit faster. Because when you're painting on a wall, the materials you're using necessitate something a little bit cleaner like that. So again, intro slide from motoring through it, but what we're doing on a wall, we are painting with acrylic. It's a beast. What are we painting or where are we painting? We are painting on a wall. We are painting on a bumpy wall. Oh God. So that's sort of like, and also, you know, when your face is 4 inches away and you're doing this thing, it's that little voice in your head is saying, why, what? Yeah, I swear I trained in this looks awful. And you step back and it looks fine. You know, it's just get a little distance. It's okay. But it is a kind of a reality. You're not working on clean paper. You're not working with infinitesimal little brushes. You're working with fast drying acrylic paint. And it's a nightmare. So, but also when you're trying to get fine detail lines, like in a dragonfly wing, you have to paint a big chunky line and then paint background paint on either side to have a little bit more of an efficient approach to making these thin lines instead of inching your way along with teeny tiny brushes on a bumpy ass wall. With the background paint, another unexpected snag was that sometimes even when you use the same paint from the same store and the same primer, like the same mix, the codes and everything match, and you get a totally different color. So that was like, okay, great. No, I'm not painting the entire wall that color this time. And this would be like a case where, yes, you do want to repaint the whole wall, which I chose not to do because I'm like, no, let's just get going. We just want to put stuff on the wall and then I'm paying for it later. I remixed it. I bought some more white paint and mixed it down so that we have a proper match, but holy hell, it's fine. And then for physically putting the insects on the wall, the first few that I did were alla prima, which is paint on wall, or I don't know what to say, French. So what this did, this is cool and it's neat, it's kind of immediate, but what it ultimately did was waste a lot of paint. Because even when you're using a specialized palette that's supposed to hydrate the paint and keep it good for a long time, a lot of what was physically on the palette dried out. And I'm looking at that and calculating, I'm going to have to buy so much paint, and I do not want to buy all this paint because I'm very poor. I went with a Kresslein method instead, upon recommendation of some of my instructors. I was like, hell. So Kresslein is grayscale. You paint in black and white primer, and you create your insect, and then you put a light glaze of color over the top so that you have your final, and then, you know, little highlights and sparkles and details or whatever over the very top at the very last stage. But the majority of the color is on one big fell swoop, which is fantastic. And this was the video that delayed our start. It is no longer a video, obviously. So it was going to show, like, in the space of 3 minutes, look, this grayscale swallowtail turns a beautiful color. It's great. So yes, the same amount, using grisaille and glazing uses exponentially less paint. So it's a much more cost efficient for this type of mural. If you had fewer insects, larger insects, absolutely go straight paint on the wall. But for this particular approach, this was the most efficient way to do it. Just pointing out, this is one of those combined enantiomorph swallowtails, so that you can see both male and female examples of the same insect on one thing, because I'm not drawing two of those. And so effectively, the same amount of yellow paint, a thumbnail-sized blob of acrylic that I used for the cicadella, was able to cover all of those other insects. Include, and then also some greens and some oranges that were elsewhere in the mural as well. So it was fantastic. I won't talk about the funding that I didn't get. I'm going to try again for one of them. But yeah, there's options to try and fund these things. And well, we tried and it didn't quite fit the requirements. But I think with the Edna Weikers Award, we can apply again with the poster version, because it will reach a wide audience, and we'll give it a shot. So all of the insects are now on the wall. They are mostly in primer, and it's just basically the flies left and then the wings. And I want to just finish with saying thank you so much to Dan and Craig of the WRC. You guys are fantastic, and I will be gone. Thank you so much.
That is stunningly gorgeous. And Jacki, are you available for commissions if people want a lab logo or a t-shirt or something like that?
After the mural's done.
Okay. All right, thank you so much. That was really spectacular. I learned a lot. Our next talk is from Peter Oboyski with a biotic survey of Sulawesi, Indonesia.
Hey, great to see everybody again in person, or at least most of you. And before I launch into this, Ernesto pointed out a lot of the challenges of doing some of this barcoding, namely that we don't have good databases that have identified material that's been barcoded. So the state of California has designated in its budget last year funding to try to barcode every species of insect in California. And I say try because, but this funding is very recent, so we didn't get a chance to put together a presentation about the project. However, we are looking for a postdoc and a museum tech with a lot of experience in both Sanger and next-gen sequencing. So if you or anybody you know is good at that sort of thing, please get in touch with Chris Grinter or I. We're both hiring a postdoc each and a lab tech each. So I'm not going to say any more about that right now. Hopefully next year we'll tell you how great we're doing on that project. So on to Sulawesi. Most of my work has been in French Polynesia and Hawaii. And I haven't spent any time at all in Indonesia. So it was a brand new project for me. And I have lots of specimens that are in the process of being processed. So this will be a data-free talk. If you're unfamiliar with the region, as I was at the time, there is the mass of Indonesia there. And Sulawesi is that funny shaped island sort of in the middle of your screen there, called the Celebes in the past, if you have older maps. And it's actually a very large area that it covers. So if you just superimpose the Indonesian islands across the US, you can see that, it's a very large area that it covers, but it's mostly islands and water. And if you just look at the island of Sulawesi itself, you can see it occupies a pretty long stretch of latitude and longitude. So it is a challenge to cover all of that space. It's probably most famous from Wallace, who, after spending time with his buddy Henry Bates in the Amazon doing the Batesian mimicry stuff, he decided he needed his own space. So he went out to Indonesia and Malaysia to start his own work there. and did a lot of collecting. He sold some of his specimens, as you probably know, lost some of his specimens in a boat fire. But he really singled out Sulawesi as a unique place, saying that there's no other example on the globe of an island so closely surrounded by other islands on every side, yet preserving such a marked individuality in its forms of life. And the reason for that is what we've come to know as Wallace's line. So you can see there's a couple of different shelves there that had these island connections over the years, but Sulawesi was never connected to any of those shelves. And so a lot of things just were not able to get there because there's a very deep channel, the Makassar Strait, which is now called Wallace's Line. And so it's a very unique fauna that's connected in very different ways. But a lot of this was based on vertebrate research. But to get at this island, where did these islands come from rather than being connected? They actually were a series, there's a lot of plate tectonics in this region, but these were individual islands, paleo islands, that over time grew and merged together. So from 20 million years ago to less than 1 million years ago, there's a lot of this merging going on until you get the final composition of that island. So what we're wondering is, do those old paleo islands still maintain some kind of signature of the fauna that evolved in those places? So there's these areas of endemism that are based on those paleo islands. And so we're trying in this project to hit each of those regions to see, well, here's the project goals right here. So test the hypotheses of these biogeographic patterns, these areas of endemism, but with more fine-grained geographical collecting and more taxa than have been looked at before. But also we're looking along elevational gradients as well. And there's a lot of agriculture, especially at the lowest elevations. You have rice fields and a lot of palm oil is taking over landscapes down there. So most of the elevation has become agriculture, but as you go up in the mountains, you get a lot of agroforestry. You've got cacao and coffee and other things like that. But they're trying to get a lot of the people out of these overcrowded cities and push them out into the agricultural areas and help with agricultural development. But that is also, eroding some of the native forests as you get higher up into those spaces. So we wanted to look at some of those gradients before they disappear. So this is a multi-taxon survey, and our PI on the project is Jim McGuire, who studies reptiles and amphibians. This is a reticulated python, the biggest snakes in the world, and the biggest ones of those are found on the island of Sulawesi and are known to eat a farmer every year or two. There's also cobras and pit vipers. And I didn't know that when I got there because I didn't have time to think about this. And my first night going into the field, just jokingly, I said, "Hey, Jim, I'm heading out into the field tonight to look for moths and spiders." He said, "I said, is there anything I should worry about?" I said, "Oh, yeah, just the pit vipers and cobras and reticulated pythons." And I thought he was joking, but he wasn't. And of course, everything I stepped on just made me start. They also have fanged frogs, not true teeth, of course, but a very cute little smile they have. We're also doing birds. So Rory Bowie is an ornithologist at Berkeley, and so that's a major part of the project, and small mammals. And so, believe it or not, these vertebrate biologists thought we should include invertebrates in the survey, which was fantastic. And Rosemary Gillespie happened to be in the room while they were talking, so spiders were included in the survey. But Rosie didn't have anybody to go do that field work. I said, "I'll go collect spiders for you, as long as I can also collect moths." And said, "Okay, that's great." So now spiders and moths are part of the project as well. Interestingly, in a lot of languages, you have butterflies, and moths are often butterflies of the night. And that's true in Indonesian as well. And kupu-kupu is the butterflies. And kupu-kupu malam is the, I didn't hear any snickering, so there's nobody who speaks Indonesian here, because butterflies of the night is also the word for prostitutes. So I had to be very careful when people would ask me, "What are you doing?" "Oh, I'm here looking for a kupu-kupu malam," and they kind of giggle and snicker. And so this is not us going there and extracting things and taking them away. It's actually a collaboration with Indonesian biologists. So these are a couple of the biologists that I've worked with in the field. And so we're sharing all the specimens. Now for the vertebrates, that's pretty easy. We say, okay, here's one of this species, one of that species. With the insects, can't really do that at the end of each trip. So they've allowed me to bring everything home to process it, and then we'll share it afterwards. And like any good project, it starts with lots of paperwork. And so here are the vertebrate biologists all being happy to sign all our permits and so forth. And they do really have a nice state-of-the-art collection down there in Indonesia. It's in Bogor, which is a little ways outside from Jakarta. And they have nice modern facilities, great collection cabinets, environmental control. So it's a really nice place to work. A lot of really friendly, knowledgeable curators there. And so, I was looking through the collection to see, I work on microlepts, and I wanted to see what they have there. And just row after row of cabinets with beautiful butterflies, gorgeous. And, some of these larger moths. And so I was looking for the microlepts, and there were 6 drawers of microlepts there. So I figured there's a little bit of work to be done. That's not to say that nobody has been doing this kind of work there. It's just not in their national collection. So I feel like this is the tortricid drawer. If you know anything about tortricids, there's more than a drawer's worth. And like every good project, you have to start with traveling. Now, as an entomologist, I'm used to bringing only what can fit on my back, right? Whatever goes in your backpack, that's what goes in the field with you. Vertebrate biologists do things a little differently. Everything you see there is us. And so it's like those old movies of, these African expeditions where you have just a long line of people carrying like tents and tables and chairs. We bring a lot of stuff with us to the field. And our trips begin by going to a village on the edge of the forest. And we do tell them ahead of time we're coming. Because we descend on these villages with about 20 or 30 people. And in this picture, you can see both the Indonesian biologists and some of the American biologists, as well as the guy who owns that house that we stayed in, all 20 of us. And we hire the locals there to be our guides, because they know their own forest, also to be porters, to bring our equipment into the fields. The vertebrate biologists carry a liquid nitrogen tank into the field with them. And we do bring a generator to charge my batteries and all that. So we hire them to be our guides and to help us with our work in the forest. And it is really some beautiful, spectacular forest where we've been able to travel. Lovely streams. This is the view out my tent one morning, looking at this nice pitcher plant right here. I mean, it's so gorgeous that even the leeches are just wonderful to look at. And you get plenty of chances to look at the leeches. Yeah. But not everything is also pleasant. This is rotan, which is the plant that your rattan furniture comes from. They take the fibers from this and so they harvest this stuff to make it grow back thicker. And those are 2 to 3 inch spines that are all over this, up the stems, on the fronds. It's everywhere. And I have a lot of physical and emotional scars from working with this plant. And then they clear a space in the forest for us. And they'll use the same material that they took down to build us a tent with tables and benches. This is vertebrate work. This is really amazing. And then we have our lights strung up, our generator running. So we have a little city in the forest when we do these projects. And what's really neat is to be in the forest with all these people doing these different kinds of collecting, different methods and bringing different organisms and all talking with each other and collaborating, helping each other out. So it's a really, really amazing experience. And so, of course, I set out lights for my moths, and I have to set up a sheet because our ornithologist will not forgive me if I don't put out a sheet. He loves scarab beetles, so he just harangues me every night until I set the sheet up. But it's a great way to get pictures of things in their natural postures as well. But of course, most of my collecting is with a bucket trap. So I have my buckets in some of these places in the forest. But now we spend 4 to 5 weeks living in a tent out in the forest, and dealing with specimens is a bit of a challenge. So for the microlepts, which I'm most interested in, I take a lot more care, and we'll do a lot of field pinning, have my Newton pins. And so I can mount a lot of those in the field that way. And I have Schmidt boxes that have 3 layers of these little pinning things that I can put about 1,000 or more microlepts in one Schmidt box, which is, that's great for letting them dry fairly quickly and getting them out of the forest safely. But the larger stuff, I just can't do that. I can't pin all that in the field. And so I have these little freezer boxes. I'll have a layer of tissue, layer of moths, layer of tissue, layer of moths. So needless to say, the DNA is probably not great in these specimens. I'm using some chlorocresol to keep things from rotting. And so, the quality is good. I'm getting some really nice specimens from them, but probably not going to be able to do anything with the DNA. So we've tried to do one trip each year, and usually 4 weeks per trip, different mountain each time we go. So we can look at these different elevation gradients on different parts of these areas of endemism. We're supposed to have our last trip in 2020. You all know how that goes. So we're hoping next summer next summer is not 2021, next summer, 2023, to finish our last trip. I get about 5,000 moths per collection for each trip, and getting those back is always fun. And of course, they're all being processed now. So I have a small team of students that's working on, pinning all these up and labeling them for me. And then, of course, there's the identification, which is why I'm here presenting to you today. If you have friends who are lepidopterists who want some challenges, I've got plenty of material that I'm never going to get to. So lots of stuff, and I will, talk to anybody who's interested in identifying stuff. And so here's just some examples of some of the things that are showing up on the sheet, some really spectacular moths that are there. My favorite are some of these things you find in these mossy forests, a lot of green moths in these mossy forests. Nice little tortricid here. This little group here, there's a radiation. I know that I've got more species than are described so far in these wonderful red and yellow things, and some day flying moths. And again, those nighttime pictures are great for seeing the natural posture. So if we spread this like a typical moth, you wouldn't see the natural posture. So getting these field photos is really essential. And so I actually managed to finish on time. So thank you very much.
Perfect. Quick question for Kate. Did you accidentally collect any beetles while you were over there?
Did I collect any beetles? Yes, I was forced to by the ornithologist to at least get the scarabs. But I was running these sheets, and I would collect any other thing that was not a lep into alcohol. So I do have that material as well. It's not nearly as much as the leps, but I do have other things, and I will post that at some point, what I have, when folks want to look at it.
All right, you guys know where to find them. All righty, our next talk is going to be presented by, are you Robert?
I'm Robert.
You're Robert. Our next talk will be presented by Robert Kresslein, moving the Universal Chalcidoidea database to a new home at TaxonWorks, the BBQ method.
All right. So, hello, entomologists and gentle friends. I'm going to talk to you a little bit about what we've been doing on the Universal Chalcidoidea database at TaxonWorks, and in particular, how we've been collaboratively approaching some curatorial and maintenance challenges. Now, let's start with the stars of the show, the best group, the Chalcidoidea. There are currently 35,796 species names associated with 27,006 valid species in some 23 families, though if you keep close on Chalcidoidea taxonomy, you'll know that that is soon to be 48 families. Most species are parasitoids of other insects or associated with galls, and there are a few phytophagous species as well. And they're very, very important in natural and biological control of insect pests. Now, the Universal Chalcidoidea database was established by this fellow right here. This is John Noyes, a research scientist at the Natural History Museum in London. And it is no longer, served online by that museum. Now, John worked tirelessly through a good chunk of his career to curate the contents of over 50,000 publications on Chalcidoidea, making sure that names and biologies were easily accessible, and many PDFs were also easily accessible. However, in March of 2019, he decided to step away from that and enjoy inserteds a little more formally, well, not formally, but a little more intensely, somehow intensely. And we needed to find a new home for this essential resource for Chalcidoidea literature and on the taxonomy, biology, and biological control with this group. And that's where we moved to TaxonWorks. Now, if some of you haven't had the pleasure of working with TaxonWorks, I've taken a moment to throw the mission statement of the project up there. TaxonWorks is an integrated web-based workbench for taxonomists and biodiversity scientists. It allows you to capture, organize, and enrich your data, share it with other collaborators, and package it for analysis and publication. It's essentially a one-stop shop for taxonomy databasing. Now, for our purposes, there are two major page types that we use. We use these nomenclature pages to collate all of the different information with names. So we have our names, synonyms and mononymies, type information associated with those names, all on pages like this. And then we have OTU pages, and this is where all of the biology goes. So we have images, asserted distributions, host records, maps, all of the citations, all connected here. And having this information separate allows us to make sure that any nomenclatural changes have the biology follows nomenclatural changes very easily. You don't have to go back every time and be like, okay, this is the valid name, let's move all the biology to the correct name. That is done automatically. There's a lot of other functionality associated with TaxonWorks. We have reference collection, object, and image databasing management. You can put type information in type images, build object matrices, and from those object matrices, produce keys. Just recently, we got the ability to do catalog output, and you can filter any of these information types as well. It's a pretty powerful tool. So these are some statistics on the UCD entities, and there's a lot to look at here. So I'm going to put some boxes around stuff that I care about. On the right here, we have all of our collection objects, over 800 collection objects, 70,000 OTUs, over 85,000 taxon names, tons of depictions, over 50,000 sources, over 500,000 citations, and over 100,000 asserted distributions and biological associations. So there's a lot in this database. It's quite large. I'm going to focus on the literature, the sources aspect of this. The TaxonWorks database of UCD contains all of the literature, or virtually all the literature, from the BMNH site. That's over 124,000 sources. And most of these publications have the PDF available for viewing. And if it doesn't, we go find it and make it available for viewing. We also have a really robust tagging system. So as we're curating this literature, we can make sure that people know what's been being processed, what's been processed, what has nomenclatural acts. And that just helps guide the different people working in TaxonWorks towards success and non-redundancy. So we get to the problem now, the crux of this talk. Last update of UCD, like I said, happened in 2019. I know it still feels to some of us like it is the early 2020. It is 3 years later. And that's 3 years of uncurated material to deal with. So without a dedicated structure or user who's being compensated to manage all of this literature, we risk, any database risks falling into low utility and obscurity, which is not what we want for this project full of love from John Noyes. So what's the solution? How do we move towards effective curation of literature? We have come towards something we call the barbecue concept. And I know you saw barbecue and we're hoping maybe there'd be brisket or, mac salad. Unfortunately, it's just data. But essentially what we're doing here is we have a small group of people working towards a common goal. In this case, it's curating papers for UCD TaxonWorks. Our model is essentially to alternate between synchronous and asynchronous sessions, with most of the time people working on their own. And you might think, well, if they're working on their own, how is that different from just asking people to go online and do work? Well, the act of doing curation in parallel encourages people to become part of a regular and consistent project. Essentially, we're asking people to sit next to each other silently and database. But if problems are encountered, we have a chat room open. We have some experienced TaxonWorks users who have been working with UCD for the last 3 years, who can help anyone who is new. So let me walk you briefly through the workflow that we do. So we start by gathering the BBQ crew on Zoom. You can see all of us up there. It's me and John Harity, Natalie Dale-Skey, Jim Woolley, and Pamela. sorry. And we all get together and start by doing updates with what's going on in UCD. So have we encountered any bugs that need to be turfed to species file group? Are there any kind of bizarre names that will take a little bit more effort? Does anyone not know how to do anything? You know, just make sure everyone's on the same page. After a brief meeting, we go and select papers. That's our second step. So this is our literature Excel sheet. And essentially what we do is we ask you to go on this and you drop your initials in that first column next to a publication. Each row contains an ID that is associated with the publication on TaxonWorks and the family that is represented within that publication. That way, experts can find their groups relatively easily and you know, specialize and do their thing. It's very helpful to have people work on the groups that they know. Things move much faster. Then you'll take that ID, jump on TaxonWorks, find your paper, apply the appropriate tags, and get curating. So as we're curating our papers, we have a priority list. We want to keep our priorities in order. And this essentially allows us to make sure that publications, any new species in those publications ends up in the database by the end of the day. And then we have tags associated with different priorities. So if we don't finish a paper, a new curator can come along and know where the last curator left off, or you know, if you forget where you left off, you have a helpful little tag there. So priority number one is to create a new name. In this case, I'm showing you the priorities associated with creating a new species. So we'll create the new species and parent taxon if necessary. A parent taxon in this context is the genus. So we associate a species name with the genus. We record the source, author, and page numbers from the publication that name is found in. Excuse me. And the original combination and rank, etymology, form, and gender of the name, and then any global identifiers like LS IDs or Zoobank IDs or anything that's in there. Priority number two is relating to holotype information. So we record the information for the holotype, the buffered collecting event, which is essentially the material examined. We just take that and dump it into a field. And then preparation type, repository, biocuration, and catalog number, any of those that are applicable for any given type or holotype. And then priority number three is everything else. So we have our paratype information. We will create an OTU page for that name, where we can associate citations with keywords, any asserted distributions, associated biology, or host associations, comparisons with other species, and any images, type or non-type, that are associated with that name. Some people choose to go through each species and do priority number one, priority number two, priority number three for the species over and over and over again. Some people will go through and do priority number one for everything in there, priority number two. But either way, this creates a framework for our curators so they know what's going on, and we have all of the information that we want in our database at the end. So we've had 10 barbecue sessions to date. Starting in May, we had our kind of pilot. Then we took a month break to refine, and then came back in July. And we've had 5 to 8 curators show up to every meeting ever since. Quick bar chart about our results. We have over 170 new species names which have been added since May 2020, which is as many species curated in the last 6 months as the combined 3 years prior. This chart I really like because it shows each of our barbecues really well. So if we look right here, can you see my mouse? Oh, look at that. You can see our first barbecue, and then you can see the next week where only superstar Jim Woolley was curating. And then we have another one with more people, and then just Jim Woolley. Jim's been really integral to getting this done as well. So many thanks to Jim. But it's really easy to see the impact of having these organized events to get curation done. We always have these little clusters, tons of people joining, and many, many taxon names added by those people. So in conclusion, since or as of November, we've curated over 150 papers with about 20, a little more papers that are currently in progress. We've caught up with nomenclatural papers through 2020 and I think 2021. So we're working on papers from 2022 now. And we have less than 500 papers which remain to be processed with 100 or so of those having no collateral acts in them. And with that, I'd like to thank you and thank all of my co-authors that are on this talk. We've all worked really tightly together to make these barbecues fun and functional. And I'd be happy to take any questions that you may have. Thank you so much.
We have time for one question.
Have you run into either electronic works that were unavailable or named available?
So
Question was.
the question was, have we run into electronic publications that were unavailable and unavailable names? I think we have access. I mean,
Yes.
okay, I was going to try to sugarcoat it. Yes, we've run into both of these. We do have the vast majority of the literature, and when we don't have a publication, obviously we can't curate it. We do have lots of people out looking for all of these publications, like I said, and usually we've been able to find them, but you know, sometimes we just can't. Yes, John.
I just want to supplement that by saying this is a great training system for editors. Because we found in a lot of cases where editors were missing things that are making them unavailable. They're forgetting to put deposits, they're forgetting to put the disability, or they miss all sorts of things. We're in the process of, we have another project authors and trying to do it. And it's been really working for something now, but we now want to, I think this is third of them trying to get it changed.
Oh yeah, that paper.
Very fun.
If you can't hear John, essentially what he said is this is a good act for finding those names. We have found many unavailable names by doing this, and we go through the process of alerting authors and helping them to right those problems.
All right, thank you so
Anyway.
much. All right, our next talk will be from ECN's very own Oliver Keller about ECN online, the lessons we learned during the pandemic.
So yeah, we're done with the nice research talks and curation talks. I'm going to take you off the highway. We're going to go back a little to March 2020, when everything changed, right? Everyone on the board for ECN was thrown into like, what are we going to do now? Like, we had to decide, are we going to have an in-person meeting? Very unlikely, or are we going to do an online meeting, right? So that then also changed what we were all supposed to do, because no one had any experience in setting up a completely meeting online. So I'm just going to talk a little bit about the good that we encountered through having, you know, this online meeting, where we increased international participation to the meetings, and we're able to include some people that otherwise would not be able to come to this meeting. The bad, of course, what did COVID do to us? Like, how did organizing a conference change? What could we not do instead? And then, not the ugly, but the future, what's going to happen, you know, for the future, like now having this kind of like hybrid meeting instead of what we had like the in-person meeting before. So I'm just going to show you a couple of quotes. I don't like really text on presentations usually, but I have to show you a couple of quotes that came from these surveys that Isabel Betancourt did after the meetings. And we had about 100, I think, people answer the survey the first year, about 50 the second. And overall, I think it was a very positive feedback of what was going on. And this one said, online brought the world closer in a very positive way. Folks who couldn't afford to attend meetings otherwise, especially enjoyed the meeting greet aspect, and the Latino session was excellent. So we had very positive feedback, collections of the world tours, international presence, and Nicole's enthusiasm and happiness. So the diversity of talks, we had the first meeting online was 2.5 days or 3 days instead of our normal 1.5 days. The overall topics, the organization, the positivity of the meeting, and then face-to-face meetings. Everyone basically said they wanted to go back to having these face-to-face meetings, but also keeping the online kind of like aspect of it to include more people. And then considering the current restrictions and limitations, I think the meetings were great and the organizers did a fantastic job on, putting this together within a half a year of where we had time to do so. And so back to like the international aspect, this is a map of the participants from the first year. We had 477 registrants, which is far more than the in-person meeting. I think they were usually around 200 or so from 28 countries. And I mean, it can, it's usually when you have the in-person meetings, you have the US, Canada, very heavy, and then, a few people from Europe join. But here we had 45 of the 50 states, and I think all but one province of Canada that were participating. We had people from, all over the world, Africa and Australia, Asia, that usually would not participate. The second year, this is the map from 2021, it went down a little bit. We only had 333 registrants, but I think that's because people were getting a little tired from all the online meetings, because I mean, every single meeting was online. I think there was a little bit of tiredness of attending all these meetings, but we still had, again, 28 countries, 39 out of 50 states, and again, all but one province, I think, from Canada. And then just to show you how this meeting is impacted, like this is the map from this year, and it shows just the in-person people. So you can see very North America heavy, a few Europeans, and one, I think, one or so from Australia. And this is the map when you include all the online people. So we have 204 registrants this year, of which 80 are online. And I think if we didn't do this hybrid meeting, we would exclude especially the South and Central American people that are participating. We wouldn't have these really cool, collections of the world, or the symposium later with from Latin America. And I think it's really important that we continue these hybrid meetings. We have 18 countries this year, 33 out of the 50 states, and 5 provinces. When you look back, I guess the superstar of the last 2 years was definitely the collections of the world. We had, I think, over 20 collections the first year, 10 or so the second, and 11 this year. And this presents us with the opportunity to see all these weird collections that we may or may not ever be able to go to. Like, all these really cool places that we might not be able to travel to. And we had very nice, I think it was very nice the first year, especially where we had collections from all over the world. And it just showed, and I think Nicole broke the record when in the first year she had over 300 people during the first session of the Collections of the World tours that were online, unique users online during the first time. And I mean, that was really a cool thing to do. And also the meetings were, if anything, much cheaper to organize. So we were able to put a little money in our account for, to save and stuff, and to use for other projects. But we're also able to give a substantial amount to our friends of the entomologists of color, and they attended the meetings. We gave them free tickets to attend the meetings, and we had a nice, amount of people joining the meetings that may have been, early career scientists, students that may not be able to otherwise attend. YouTube. So also we were able to this time film all these videos. We had to process them, but they're all, the last 2 years are now all available on YouTube. If you want to subscribe, all the videos from this year will also be made available. I can't tell you when, but they will be made available. And now I'm going to switch a little bit to the bad. So why do we come to the meeting, right? We want to mingle, we want to socialize, and we want to talk to our colleagues that we haven't seen all year. So this is it. But 2020 happened. No, we're not doing that. What did we do instead? We saw each other all on Zoom, right? Which some people said they liked the breakout rooms. A lot of people said they didn't like them. I mean, that's just like it. It can be complicated if you have breakout rooms that are random. You put people in a room that may not know each other. It's hard to make a conversation. You get lucky, you are in a group with people it's easier to make a conversation. So, but a lot of people just said, thank you to making the virtual meeting, and that they couldn't attend otherwise. I mean, first year, that would have been probably 300 people that would have not been able to attend the meeting. Organizational, it changed. We had, I think, very well-defined rules. So I wasn't on the board for the last in-person meeting. I was kind of thrown into the whole virtual world. I think online we have very well-defined rules, what everyone does. The president, talks to the sponsors, gets the sponsors together and stuff. The vice president organizes on site with ESA, gets us the room, blah, blah, blah. It's, everyone knows what they're doing. COVID happens and we were all scrambling. Like Nicole was the vice president. She was way more involved in, getting the program together, writing these texts to instruct people what to do during the videos, how to record the videos, how to make subtitles. Her and Jennifer were involved in that, and Karen Wright was also very much involved in that. I stayed with my program, but Chris was the president. He still talked to the sponsors, but on the day of the conferences, he was sitting at Calicat with his computer plugged in the hardline and, making sure that all the videos were playing without problem, not being on a Wi-Fi connection where all of a sudden the talk is gone and whatever. So the roles very much changed, I'd say. Maybe treasurer and secretary, not as much maybe, but everyone else was like really scrambling and trying to figure out what are we doing and how are we doing it. For me, program chair at that point, it was nice. We could make the program until the day off. We could make changes. We could add people. We could change the abstracts. This year, the program has to be done 3 weeks beforehand, right? And I have to send it to the printer. So far less easy to change things. So the whole organizational thing was really a time sink. Like it was really a lot of work. I think a lot more work than usual. So yeah. There were some bad things, but I think we got it done. And then the second year we had a little more, we knew what we were doing. And it was a little easier to do it. And also it prepared us kind of for the hybrid meeting because, we have the instructions now to send out to the collections people, this is how you make your video, this is how you add your subtitles. Then it's YouTube. It's also the bad thing because, it took us forever to take all the videos, cut them, make them available, and you are after the conference, you have that like little, your motivation goes down a little. So maybe that, contributes to taking a little longer to get the videos online. Otherwise, so this is the future. We have to do these hybrid meetings. We have to figure out how we're doing them. And this year is I think, good run. There's a few things we need to tweak, but I think that's the future. We have to include and should include people from other places to be able to attend. And I want to thank everyone that was on the board at that time, Chris as the president, Nicole, vice president, Christy and Patrick, Karen Wright, Isabel and Ashley. They did a great job during that time, and it was a struggle, but I think we got it done, and I think we learned a lot from those 2 years. Thank you.
Thank you so much, Oliver. That was fantastic. I just want to say thanks again to the members of the board who have all worked incredibly hard to pull this off. I've never actually seen a successful hybrid meeting functioning with concurrent virtual and in-person speakers, and I'm absolutely stoked. And a round of applause as well for Andrew, who's helped us actually pull all of this off. Kept the AV running, which is not a small feat, and I sure couldn't do it myself. Our next talk is entitled, "Why Are There So Few Lepidoptera Type Specimen Catalogs?" and will be presented to us by Geoff Martin.
And Natalie.
All right. Oh, your tag team.
Yeah. Okay, morning all. I'm Geoff Martin, Senior Curator of Lepidoptera at the Natural Museum in London. This is Natalie Dale-Skey, the Senior Curator of Hymenoptera in London. And we're going to try and answer the question, why there are so few Lepidoptera type specimen catalogues. The findings here may well apply to other insect orders, but that's beyond the scope of my knowledge and this presentation. Okay, so as I'm sure everybody knows here, type specimens are fundamental to taxonomic studies, and improving access to type material should be a priority for type specimen depositories, yet type catalogues are surprisingly scarce.
Type catalogues that do exist tend to cover a certain author, historical authors, taxonomic groups, and a very few entire institutional holdings. So why are type catalogues needed? Certainly the NHM UK and no doubt elsewhere, there is a huge demand for digital images of type specimens. Personally, I get a request a week for digital images and I'm only responsible for the Geometroidea. Many people also assume we have all the types, and in fact, where they're housed elsewhere, and also they're not really aware that it could be more than one primary type for a given taxa. Or, in fact, that the circular BM type label may in fact be incorrect. Digital repatriation is a very important issue now. For giving access to type specimens, visiting collections is often beyond the means of many researchers, and the majority of Lepidoptera types are held by 3 institutes, London, Paris, and Washington, whereas most biodiversity is in tropical countries. And there's a taxonomic impediment without access to types. Obviously, physical loans are available, but certainly the policy at the NHM UK is, which is usually adhered to, is 6 types, primary types per loan, and for a maximum of 6 months. And of course, there is a risk of sending the types out in the post that they may be damaged or even worse, lost completely. And also, there's not actually very many type specimens of deemed sequenced. So obviously, it's very important you are, when you're sequencing type specimens, you are actually sequencing the type. So if you do a quick search on a Lepidoptera type catalogue in Google, surprisingly, you don't get anything particularly. The first hit is for a generic names and moths of the world catalogue produced by the museum, which isn't the type catalogue at all. There's a couple of references to type catalogues on BHL, both of which are quite old. Then the first one you come across that's possibly of any use is the type catalogues of the Museum Koenig in Bonn, which again is just a list of the types they have, just the name and the type status. And then the first one that you come to is number 5 on the list that I think is what a type catalogue should look like, is the Zoological Museum in Hamburg. This is available in Evolution Systematics, published in 2021, this particular one, which is the illustrated catalogue of type specimens of the Papilionidae. It's an open access journal, but it's also downloadable as a PDF. So you've got, you know, the modern and the sort of more traditional method of having this type catalogue to hand, authored by Zahiri et al. And for me, I think this is what we should be aiming for. It's got hierarchical search links and there's clear information and remarks on the type status where necessary. So I'm going to quickly look at the types online and what I call the big 3. That's the Muséum National Histoire Nationale in Paris, the USNM, Smithsonian, and the NHM UK. And this is not meant to be a critique of anyone's institute, it's just a quick look to see what is actually available. So if we look at the USNM, their website states 25,000 primary types. There's a nice hierarchical drop-down list, which is very easy to use. All their types appear to be in the database. Not all of them got an illustration, got a color image, but here's an example of one. And also the original reference is also quoted, but there doesn't appear to be any type verification as such, because some of the type statuses are just listed as type. Then we move on to the Paris Museum, 11,000 primary types, very nice images, but only 3,619 appear to have a record, but they all have images. Obviously, this looks like it's work in progress. Good search engine, again, hierarchical, but no verification of the type specimens. And then we come to my institute, 125,000 primary types, but only 3,712 types appear to have a record. All of them have images. The search engine is not hierarchical, which I don't particularly like. And again, no verification of the actual type status. And so the big 3 quick conclusions. The image quality of all 3 I thought was excellent. Image available for every taxon listed. Only one institute seemed to provide that. The data was downloadable on all 3 as a CSV file. The list of complete holdings, only one institute had that. Type verification, 0 institutes appeared to do that. And in my opinion, I like a nice hierarchical search engine, only 2 out of 3 provided that. So I'll bring you on to the Madagascar type digitization project. In 2017, the John Franks Trust gave us some money for a 2-month pilot digitization project. to image all the type specimens, primary and secondary, from Madagascar. So 2 digitizers are employed by the project, and we use the Afro-Moth checklist, thanks to Willie de Prins, who managed to filter this for us on Madagascar and B&MH type depository. And then we match this with our own collections index that locate the actual specimens. And the types in our collection are often labeled with a little round BM type labels next to them, so they're quite easy to find. So in the 2 months, 2,600 specimens were imaged, but this also threw up a whole host of issues we hadn't even considered. There is no separate type collection, so we've got a collection of only 80,000 drawers on 4 floors. So obviously, if you're looking for specimens, this action alone, just finding the specimens and taking them back to the photo station, took 27% of the digitizer's time. So a quarter of the time was wasted spent doing that. And we also found the Afro-Moth checklist had many missing, the type depository was missing in quite a few places. And our collections index also left a little bit to be desired for some groups as well. So where do we go from here? So obviously, clearly we need a revised workflow. So we managed to get more funding from a government department called BASE in 2018, and we got the digitizers again for a further 3 months this time. At this point, the Lepidoptera curators, that's myself, Alberto Zilli, David Lees, and Alessandro Giusti, asked ourselves the question, how hard can it be to produce a type catalogue now we've got all these wonderful images? So we decided this time that the curators, us four, were going to locate the specimens, check the original references, and actually build a separate type collection before the digitization takes place. So that basically there was just one place in the collection where the specimens are stored pre-imaging. So after 3 months, we've now got 5,825 specimens have been digitized, and we are attempting to produce a type catalogue sometime soon. Right, I'm going to pass over to Natalie now. We've dealt with the data, and I'll be back in a minute.
Yes, hi everyone. I'm going to talk about some data aspects of the project. So as Geoff mentioned, our data set is just under 6,000 specimens covering just under 1,500 taxa. 70% of those, for 70% of those, the original description is available in a BHL. That's just to highlight what a fantastic resource this is. And the number of type specimens at the BM per taxon goes from 1 to 354. So in fact, the vast majority, 925 taxa, there's only 1 specimen at the BM. Most of them cluster in the low number of specimens. And there's a few outliers with very high numbers, clearly authors who thought you can never have too many paratypes. And to give you an idea of when the names were published on the graph, the height of the bubble indicates the number of publications in any 10-year period. And the size of the bubble, as well as the number, is for the number of names in that period. So you can see a high number of names and publications in the late 19th century, and again, a big cluster pre-1950. And that's not necessarily good news. And it's not good news due to issues linked with typification. So assessing the type status of specimens is clearly going to be a large part of producing a type catalog. And the two questions you're asking yourself is what kind of type was fixed for a taxon? So for instance, a unique holotype or a series of syntypes. And is this specimen in my collection part of the type series? Now, in the current edition of the code, there is a requirement for a name to be available. There must be an explicit fixation of the name-bearing types in the original description. And so you should, and in fact, you must get the answers to both questions from the original description and neotype lectotype designation if relevant. But for all the names, there was no such requirement, and the information on type specimens is often lacking. For very old names, is lacking most of the time. So one of the big issues we often face is when there's no information on the type specimen, and you have to decide, was it based on a single holotype or a series of syntypes? And that is that most of the time it's open to interpretation. So in the code, there is a recommendation that if there is no explicit or implied fixation of a holotype, you should err on the side of caution and assume it's a syntypic series, but it's only a recommendation. And to decide whether any specimen is part of the type series, you can use published and unpublished evidence. So if it could be labels, notebooks, correspondence. And if you are a bit obsessive, I'm not suggesting that curators tend to be, but you could potentially spend the rest of your life looking for that one piece of evidence that demonstrates your specimen is a type. So the main message is typification is not always clear-cut, especially for all the names. And the best thing you can do is accumulate evidence if you want. And crucially, you should document this evidence. So obviously the team was not gathering data just on type status. By the time I joined the project, the digitization phase was over and a lot of information had already been collected. And it was all in a spreadsheet. And this is basically why I joined the project. I am not a lepidopterist, but I love Excel. And at the time, we were in lockdown. So we had no access to the collection. So any computer-based project, which was a little bit different, suddenly became very appealing. So I suggested to the team, as you're working on a spreadsheet, why don't I create a data entry form? It's going to be much easier to use than working directly on the spreadsheet. And this is the form we are currently using. So I could bore you to death describing all the functionalities of this, but I will resist the temptation. And believe me, I could. And just highlight that obviously one of the main advantages is you don't have to scroll along the spreadsheet. You can see all your fields in one go, and there's no risk of entering data in the wrong row. And I just draw your attention to the little checkboxes on the right, which we added at a late stage in the project when we decided to look at to record which BM type labels were present on the specimens. So these are the labels I'm talking about. These labels are found on a lot, not all the material, but a lot of the type material at the BM. And we decided to look at those because we are aware of the fact that a lot of users of the collection tend to assume that these are completely trustworthy, so equivalent to a verified type status. Sorry, I'm completely out of breath. And in fact, we know that's not the case, but we didn't know how bad it is. So we thought we're going to look at this. So it's preliminary data. Because we started doing this at a late stage, not all specimens have been assessed, and a lot of things need checking and need more scrutiny. But out of 2,054 specimens, more than half have no BM type labels. So they might have other labels. We're just looking at those circular ones. 281 have a type label as shown on the left, which just says type, which is potentially not hugely useful. But in all cases but one, these are found on either a singleton in the collection, or when it's a specimen part of a series, only one specimen out of the series does have that label. So this, together with the fact that type was used a lot as equivalent to holotype in all publications, we, at least for now, we assume that this label means a unique name-bearing type. So are these labels reliable? So out of 905, 694 were correct. So the type status on the label is the same as the type status as assessed in the project, which I must say is a lot better than I thought it was going to be. I'm not going to go into all the other ones because, as I said, it's preliminary data, but just to highlight what seems to be the main issues. So you have 149 unique type labels found on scene types. Now, if you look at those, 80 of those were assessed as in the project were assessed as scene types through the application of a recommendation 73F. So that's the recommendation that if you have no indication either way, you should be cautious and assume your specimen is a scene type. But it's not incorrect to throw caution to the wind, and you could have good reason to think it's a holotype. So these are not incorrect, but that still leaves 69 specimens labeled as unique types, which are part of a type series. Either we have more types at the BM or the original description is clearly based on more than one specimen. Now, one possible explanation is that all those tags are pre-1939. And we know that at the beginning of World War II, a lot of the type material was taken away from the museum for protection. And apparently, when there was a type series, only one specimen was taken. So potentially, they could have been at this point labeled as a type, but obviously that needs more research. And paratype or allotype labels on syntype, it's a linked issue. It's a mixture, it's a mix basically between name-bearing and non-name-bearing types. So based on this preliminary data, the main issue seems to be certain specimens have been segregated and labeled as a unique name-bearing type when they're in fact part of a syntypic series. And well, certainly people should be made aware of this. And that's it from me.
No, there's more. Don't get away from it. So I'm going to go talk about type verification. So in what we're looking for, the ideal species description will have a list of all the type material of the depository. Specimens will be labeled as types and hopefully and preferably unique specimen numbers, color images, and published in an open access journal. We found no example of this in this data set. And missing items above make type verification uncertain and or difficult, and of course, much more interesting. And below is a typical Walker specimen with a description next to it. And we're going to assume that's a syntype for now. Okay, I'm going to run through a few issues briefly. So Natalie just mentioned no indication of number of specimens, holotype, or is it a syntype? And again, you see here the description, helpfully in Latin, but certainly there is no indication of numbers of specimens, although the specimen here does have a holotype label. That's a decision we need to make on the basis of the evidence, whether that is a holotype, but in fact, this could be more than one specimen in this series. And another issue we come across is the syntypes are unlabeled as well. We see here this specimen, the description says 5 males and 1 female. We found 5 males and 1 female, but only 2 of them have the museum type label on. And then another issue was the syntypes unlabeled and of an unknown number. Prout in particular just used to say a long series. I'll show you more here in a minute. So we found 35 syntypes. We believe there are no more. But if you look at the specimen on the left-hand side, it has the type label, but it might not be the type because the date is not as in the original description. So maybe someone's a bit hasty putting that type label on. And we could play Prout's type series bingo. So here's an example. I'm going to give the numbers away here, but so about 20, 17, several of the males were 7, a short series, 17, a series, 28, a good series, 32, a long series, 44, a long series of males, 75, many males, 28, and a very long series, only one more than a long series, 76. So, and do these minor errors invalidate type status? That's something we need to think about. Another Prout specimen, we're not picking on Prout, I mean, he was a good guy. So we have the specimen is labeled as type and paratype, but we look closely, the type label specimen has no date. Are we going to worry about that so much? And the second specimen labeled as a paratype is from the Joycey collection and not from the Trim collection. Now, obviously, people exchange specimens all the time, particularly paratypes. And there's not really much of a record of where they've gone. So we need to make a decision whether they are part of the syntype series or not. And there's 2 further specimens at the bottom. Again, we've got the wrong date. How picky are we going to be? Now we come to another issue now. Obviously, with these big syntypic series, it's always very useful to have a lectotype designated. Although labeling it is probably quite a good idea as well. Now, this is a fairly good example of there was a catalogue produced on the types of the bee, just the moths, by Viette and Fletcher in 1968. So if you look at the original description on the right-hand side, you see there's a male and a female mentioned, no numbers. And then the lectotype designation, the lectotype's been designated and labelled and published, and there's one paralectotype, but they're both females. So where the males have disappeared to, well, we're not quite sure. Or another challenge is when the lectotype is unlabeled. I mean, how are you going to find out there's been a change in type status? I mean, you know, obviously access to literature is important here. So if you look on the left-hand side, the original description is there, 2 males from Madagascar. Both the males are there. And then in 2001, Kruger decided to designate it a lectotype. I mean, obviously, he never told us he was going to do this. Obviously, we found it in the publication. And then the paralectotype is listed with the same data as the holotype. So again, all these little minor errors are creeping in from the past and the very near present. And then we have something called lectotype limbo. So the original description here is brilliant. Specimens all bad. It's not beat around the bush. Males and females are listed. We found 5 males in the collection. The female appears to be missing. And if you notice, I've just added this in late point. The type description, the type locality is not listed here. Just at the beginning of the article, the type description says, thence work to a place rejoicing in the name of Nalama, I shouldn't even bother with that, should I? So 2,000 feet above sea level, etc., etc. And it says almost all the insects were found there. So what does that mean? I'm not quite sure. So anyway, we've got one specimen label with the red type label again. And if we go to in 1951, Viet did a work on what was a costed moth then, and designate the lectotype then, but he says it's a male. And of course, because all the 5 specimens have all got exactly the same data, he didn't say which one. And unfortunately, his illustration on the right-hand side, that specimen there, which is figure 6, I think, is not particularly good. It doesn't match any of the 5 specimens. So he may have used one of his own specimens to illustrate the fact that it's an example of that species. So what do we do with that? Don't know yet. So I'm going to round up now. So the obstacles of producing a type catalogue. Access to literature is one of the biggest ones. Obviously, 70% of the literature we found on BHL. I'm assuming we'd find the other 30% across the corridor from Mike Farrow's office, but you never know. Yeah. And as you see, many of the descriptions are poor, not illustrated, and are a bit of a nightmare to work out what's going on. Time is another factor. And the Museum Histoire Naturelle de Geneva just produced a type catalogue, and it took them 14 years for 437 specimens. On the basis of that work rate, we reckon we might have finished in 188 years' time. Curatorial skills, very important. I mean, the knowledge of the collection you're working on, and hopefully someone's got the knowledge of the code. And by the time we finish this, I think we'll all be experts. And another issue was the types not being separated from the main collection. Obviously, this is a big discussion museums have. Do we keep the types separate? Do we mix them with the general collection? I always used to be in favour in the general collection. Having just worked on this small data set here, I'm dead against having types in the main collection now. They should all be separated, particularly if you want to digitize them or sequence them. And I'm just going to finish on this beauty. So this is probably the most complicated one we came across so far. So this is one specimen, and it's the type of 2 species. So I asked David Lees what on earth is going on here, and I hope I've got this right. So I'll start at the beginning, and if you want to throw me off the stage, please do. So this was described in 1867 by Felder and Felder. That's a father and son combo, and that's probably a talk in its own right as to which Felder is which. And the name of Icthema batesii, and this specimen is the holotype of that name. And in 1885, Mabie published a plates volume in volume 19 for his work in Histoire Naturelle de Lepidoptera, which is in a big volume grandidier in Histoire Physique Naturelle et Politique de Madagascar, which included 4 paintings representing 2 specimens, not this one, of what he calls Strabina coronites. And funnily enough, coronites is currently regarded as a junior scientific synonym of Icthema niveata, Butler 1879, but we haven't got time to deal with that. And then in the corresponding volume 18, which came out a year later, so 19 came out before 18, published in 1886, published in 1887, although the frontispiece says 1886, he actually refers to this specimen described by Felder as being part of the series in between the 2 names. So this might explain why it's got 17 labels and why type catalogues take an exceedingly long time to do. So on that basis, there's a few acknowledgements and any questions?
Do we have any questions? Yes, Margaret.
Separating sites versus not separating sites. It seems as though there's actually quite a good evidence. In separating them should only be done after you've done all this site verification. Otherwise, you can, let's say, what's labeled those sites and pull it out. You may not know that there's more than one.
Yeah.
Repeat that for the,
So I was asked a question, you should really check the type status sites before separating them into a type collection, which I agree with, because as I say, obviously you just don't know, particularly with older material, what is going on until you really look at it in detail.
all right. We have one more question.
Just to point out, I'm not seeing that people have a couple of talks and presentations here refer to allotypes. I hope people here realize that allotypes are not recognizing the ECN at all. They are parent. All they are, that's all everything should be lumped together. It's all they are parent. Not be contained or labeled separate from parent.
Yeah, totally agree. But when we come, sorry, there was a question there about allotypes as not being correct in the code, and they are nothing more than paratypes. I'll just say I agree with that. But certainly when we come to produce this catalogue in 188 years' time, we will mention the fact that it was described as an allotype, and then there'll be current type status, which will be a paratype.
Just to comment on that, the reason I still think allotype, even though in a code they are not the nomenclature is significant. I know if there's a long series of males when the holotype is female, the allotype for me is the specimen that whoever described the taxon, he thought was most representative of the taxon. So I think they do have a little bit of significance.